All right. Our next speaker is going to be Dr. Crystal Hepp from Northern Arizona University, presenting on genomic epidemiology or assess environmental risk. Um, Dr. Hepp, if you want to go ahead and unmute yourself and turn on your camera.
Yes. Can you
Cool.
see me?
Yes, I'll hand it over to
Okay,
you.
great. So I think it was supposed to be genomic epidemiology to assess environmental risk. But give me a second here, and I'm going to share my screen. Let's see. Okay, are you able to see the screen, the little map moving around?
Yes.
Great, thank you. All right, thank you. I appreciate the offer to come and present at this conference, and just want to give you a little bit of background on my lab and the work that we've been doing, and how we intersect with public health partners. So again, my name is Crystal Hepp, and I'm both at Northern Arizona University. That's 75% of my time, and then 25% of my time at the Pathogen and Microbiome Division of TGen. So we call that TGen North up here in Flagstaff. My group really got into environmental surveillance for viral pathogens back in 2016. Some of you might remember that during that time there was a global Zika outbreak, and there was a big concern about Zika being spread into the United States, including Arizona. So of course I thought that I would just start doing surveillance for Zika and contacted Maricopa County Vector Control. And they said, we are getting Zika funding, but the thing that's really a problem for Arizona is West Nile virus. And from a genomic perspective, there aren't any people in Arizona really looking at that. So that really, listening to what our public health partners had to say and the priorities that they had is really what my career has been based on, and it's really developed in that way. So we'll be talking about West Nile virus, as well as some other viruses that we take a look at. So one thing is, so my group has been working with Maricopa County since 2016, and how it goes is they have approximately 800 traps that are set out through the entire county every week. Um, Mosquito traps, carbon dioxide based. And then after the mosquitoes fly into those traps are usually set for about 12 hours. Somebody comes back, they pick up the traps, they take the mosquitoes from the traps back to the lab and separate them by species and sex. And then they test them all for West Nile virus, St. Louis encephalitis virus, Zika, the appropriate species. So Aedes aegypti would also be tested for Zika, chikungunya, and dengue. Um, and then after they've tested, what we do is we focus on the positive West Nile virus mosquito pools, and SLE positive mosquito pools, but we do take negatives as well, for comparison purposes. We bring those pools back up to our labs in Flagstaff, Arizona, and then we do genomics on those. And so that's what I'll be talking about. The first part of the project, the first part of the talk is the genomics that we carry out. This map that I'm showing you right here is a little bit of a teaser. So what you can see, we have several partners throughout the Western United States through the Pacific Southwestern CDC Center of Excellence for Vector-Borne Diseases that I co-lead. It's based out of UC Davis, but Arizona has a pretty significant imprint within that CDC Center of Excellence. And so what you're seeing on this map are different points where West Nile virus positive mosquito pools were collected. And the genomic information allows us to make connections between those different locations. So you can see here, what we're finding is we have in this middle area, we have a lot of red, and that's Maricopa County positive West Nile traps. Then we've got Yuma County in maroon. We've got Riverside County, California in the lighter blue. Las Vegas, Clark County is in the purple, lighter purple, and then St. George, Utah is in the darker purple, and we've got Flagstaff in the orange. But you can really see that Maricopa County sits in the middle of a lot of these different transmission networks. And so our feeling is that putting a heavy focus on Maricopa County probably has a bigger impact across the entire southwestern United States. And just to zoom in a little bit, again, on the left-hand side, you can see the same map with slightly different coloring, but then on the right-hand side, you can see that we're also doing a surveillance in Salt Lake City, Utah, and they are also interested in trying to understand within their area how is the virus moving? Are we able to identify source locations? And we'll go into that a little bit more here. Okay. Maybe I like virtual is always hard for the interaction purposes, but can I get a thumbs up from anybody that works on West Nile virus related surveillance or mitigation activities, response, that sort of thing? Okay, not seeing a whole lot, but possibly I just can't see it on my screen when I'm sharing. But so for West Nile virus, what we know is that it primarily circulates between mosquitoes and birds. And every once in a while, you'll get transmission into human or horse or some other dead-end host. And it's a dead-end host because that host isn't able to develop a high enough level of virus in their blood such that an uninfected mosquito could come by, take a blood meal from them, and then get infected themselves. But birds, they develop a really, really high level of viremia. And so that's what allows the cycle to perpetuate, because then an uninfected mosquito can come along, bite an infected bird, and that keeps the cycle going. And in much of Arizona, what we know is that the two big vector species for West Nile virus, on the right-hand side, we've got the Culex quincofasciatus and Culex quinc for short. And then we also have Culex tarsalis on the left-hand side. I think tarsalis is better looking just with the little striping on its legs. And so we can get virus information from these mosquitoes. We can get mosquito genetics to understand how mosquitoes are moving over the landscape. And then also you look at this Culex quinc on the right-hand side and you see that there's blood in its belly from the blood meals that it takes. And so we can also sequence use sequencing to identify what host that mosquito has taken a blood meal from. So all these birds that you see around the cycle picture are birds that we have found that mosquitoes in Maricopa County are taking blood meals from. So there's quite a few of them. Another thing that we're trying to understand right now is if birds, bird blood meals that are St. Louis encephalitis virus positive versus West Nile virus positive, we want to know if there's a difference in the birds that are being bitten, transmitting those two viruses. West Nile virus initially entered the United States in probably in 1998, but was first detected in New York in 1999. And what you can see is that because there were competent vectors across the entire United States, all Culex species mosquitoes, that the virus really was able to make it across the United States pretty quickly, which is, a collaboration between birds moving the virus as well as the vector being present. And if you focus, hopefully you can see my pointer, but if you focus on Maricopa County, from here forward, you will see that the county never turns white, meaning that we always have non-human activity, which is usually a mosquito positive pool, sometimes birds. But we always have non-human activity, human infections, or a combination of both. Again, I'll keep my cursor there. So you can see over time, we just are constantly having West Nile virus, despite the fact that the virus moves throughout the rest of the country a little sporadically, it seems. What we also know is that we can compare the case counts, the human case counts in green, to the number of mosquito pools over time. And you can see that there's a pretty good correlation indicating that the trapping that's being done in Maricopa County, it really is reflective of the number of human cases over time and space. So very strong surveillance strategy. And so that means that environmental load of West Nile virus is useful for predicting spread into humans. So our work really focuses on trying to understand what locations can support overwintering and endemicity of West Nile virus. So we want to know which locations the virus can stay at during the winter time and just reemerge, allowing it to be an endemic population, just staying within that area, versus locations where there are importations that have to seed into the county, a different county each year, to perpetuate a new West Nile virus cycle. So where does the virus overwinter? We also want to know, are there hotspots in Maricopa County that can be considered for proactive mitigation activities? If the virus is just randomly circulating, it's really hard to think about the most important locations to treat. But if we can identify a few hotspots that might be really important for overall spread, that allows potentially better targeting of resources. Can we identify better times that we should be triggering a public health and community response? We think that we might be able to do that using genomics as well. And then what additional surveillance methods could be useful? And so we'll talk a little bit about wastewater-based testing as well. Our lab, again, we have funding through that CDC Center of Excellence for Vector-Borne Disease. And we have been able to sequence more than, I believe, 3,000 West Nile virus genomes from the Western United States. And one thing that I wanted to point out is that, so there's a lot of different colors going on here, but you can see in the maroon is where we have most of Arizona, and the vast majority of those samples are from Maricopa County. But you can also see that nested inside of the maroon color, we see blue and we see pink. And what that means is that those are locations that Maricopa County is contributing to. What we can also do, so we can say, okay, this right here, based on the timeline, is a 2024 sample. And if we were to map backwards over time, we can see when this particular clade entered Maricopa County. And so this dominant clade that circulated for a really long time actually was initially seeded in about 2012 and lasted until about 2024. Or 2023 and 2024. And so what's really interesting about that is that in 2023, we had a really, really hot summer. And then after that, we saw this new little clade emerging, this Maricopa County endemic population 3. And so you can see we have samples from 2023 and 2024. So it seems like the heat really knocked down the old endemic West Nile virus population and allowed for seeding of a closely related but distinct population of West Nile virus. And that new population does have some distinct amino acid mutations that could affect the virulence of the virus. Another thing I want to point out, on the right-hand side, you can see the flyways that we have, the general flyways that we have in the United States. And both Arizona and Utah are located in the Pacific Flyway. And if we think, well, we know that birds are the primary movers of West Nile virus over long spaces. So one would think that Utah and Arizona might have closely related West Nile virus strains. But actually what we see is that, so Utah, this is from the Salt Lake City Mosquito Abatement District, most of these samples, and they cluster very far away from the Maricopa County samples. And in fact, what we can do is we can draw back to the time where they connect to each other, which is this big long branch here. And that was approximately in 2001. So the strain of West Nile virus circulating in Utah has not seen the strain circulating in Arizona since the very beginning of the outbreak of West Nile virus in the United States. And that could be because they have two distinct vector species, and perhaps those, so up in Utah, the primary species is Culex pipiens. And so those two different vector species could be driving the differences that we're seeing in strains, and perhaps we wonder if it could explain differences in, for example, West Nile virus neuroinvasive disease. Okay, so again, why focus so heavily on Maricopa County? I brought this up once, but again, if you look at Maricopa County and how it is connected to other parts of the southwestern United States, you can really see that it's more of a centralized location, what we might call a source location, because we do see that it ends up seeding a lot of other locations in the southwestern United States. And what you're seeing here is sort of a snapshot, but there are many other locations that Maricopa County is connected to. The other thing that we can do after we build these phylogenetic trees is we can take the tree, like you're seeing on the left-hand side, And we can build a network out of this. And people always laugh because I sort of say that this network here is what really clarifies everything, even though I do realize that it's initially pretty complex to look at. But as I mentioned earlier, Maricopa County has 800 traps distributed throughout the county that they test every single week. So each one of these bubbles that you see is an individual mosquito trap. And so because the traps don't move a whole lot, every individual mosquito trap represents an individual location within the county. And the larger the bubble is, the more connections that location has to other locations within the county. So you can see there are a lot of larger bubbles, but there are even more really, really small bubbles, and you can see that they just have one connection. So this trap in green that has many connections leads into several dead-end sites that don't have any other connectors. And if we just put this into a histogram that's a little bit more easy to interpret, what we can say is that there are a whole lot of trap locations in Maricopa County that are that have very few connections. So maybe they're only connected to one other trap site, but there are very few traps that have a whole lot of connections. And that's exciting because it means that there are only a few locations within the county that are really responsible for perpetuating West Nile virus over the long term. So this is kind of a big picture example, but we can also, going back to this, sorry, these are all of the dead end sites. So these sites are likely not as important for ongoing West Nile virus transmission, or it could also mean that the mitigation activities that are being carried out by the county get to that trap site so quickly that it drives down the virus before the virus is allowed to spread. So it could be one of those two situations. So we have a few different clades that are clusters of West Nile virus that we have zoomed in on. And we have this one cluster that we call cluster 3. And cluster 3, you can see down here, is composed of different West Nile virus genomes that were present in 2021. And then these, which I'll talk about in a second, they're all present in 2023. And if we look at them, look at how they're on the map, they're kind of all over the place, but you really do see that there's a lot more red right around this particular location, and closer transmission traps that are closer together are more likely to transmit to each other. And if we break this down into that network, a very small network, what we can see is there's a trap called RT262, and that trap is connected to 5 other trap locations. And then, of course, RT254 is connected to 2 other locations. It's connected to RT262 and then RT305. And then we have several dead-end sites. This trap, RT262, and we're looking at 2021 strains, is here in this particular location in Maricopa County. So there's a lot of water, there's a lot of birds. It's a really good location for everything that's needed for the cycle to be perpetuated to keep going. But then we can also carry out multi-year analyses. So now we're adding in our 2023 strains, and we can see that this RT262 trap is directly connected to itself, RT262, but in 2023. And so what this connection indicates is that this is a good location, not just for the endemic cycle within a year, but also endemic cycles across years. It indicates that this is likely a source location where overwintering is possible. Now, I will say we talked with Maricopa County about this particular location, and based on their own research on their data, and then coupled with this data as well, they were able to talk to one agency that owns some property in the area, and get some appropriate treatment put into the area. And this location is no longer as big of an issue. It's not as problematic as it used to be. So that's sort of one outcome from this effort that we're pretty proud of. What we can also do with the phylogenetic information is we can estimate something that's called effective population size. And that just sounds really, really complex, but actually what it is, it's just a comparison of how much diversity there is in the viruses that are in Maricopa County mosquitoes. And by diversity, I mean number of mutations. So there are a whole lot of different strains or different variants that are present at any one time, or there could be more or less depending on the analysis. So you're going to see this median effective population size number, that's going to be in blue. The number of human cases is going to be in orange, and then the number of West Nile virus positive mosquito pools, that's going to be in purple. And you can see that the purple and the orange, they do a pretty good job tracking each other in the very beginning of the 2021 outbreak. But later on, what you see is that as the human cases rise and rise and rise and rise, you do see sort of a fall off of the mosquito count. And I think that can probably be attributed to how mosquitoes are pooled. So within a particular trap, if you have 50 mosquitoes, all 50 of those mosquitoes will be put into one single mosquito pool. And then it's just counted as one positive mosquito pool, even if you might have 20 or 30 positive mosquitoes present. And so eventually the signal from the mosquito pool can get saturated. But with the genomics, what you can see is it keeps growing and growing and growing. And the environmental diversity starts rising before the number of human cases starts rising by about 2 weeks. And then you can see it just keeps increasing over time. And this difference that you see right here is because we didn't sequence as many genomes in this particular time frame. But what this indicates to us is that genomic epidemiology could possibly assist in triggering public health and community responses if that's something that public health wants to build in. And then I said that we would talk about additional surveillance strategies that can be used for West Nile virus and other arboviruses. Our team started as many other teams did, in March of 2020 during the COVID pandemic, when the COVID pandemic was really kicking off. We started working with our City of Flagstaff collaborators to test for SARS-CoV-2, and then eventually built into quite a large panel that we run every week for some different collaborators. And one of those collaborators is the City of Tempe. So because of the big 2021 outbreak, City of Tempe, in addition to the respiratory and gastrointestinal viruses that they wanted to surveil for, they also asked if it would be possible for us to carry out surveillance for arboviruses at their different wastewater collection sites. So we started doing that. So we look for West Nile, SLE, dengue, chick, and Zika. And then what we found is that in 2023, there were 10 detections. We found there was one detection in July of 2024, and we have had a couple of detections now in 2025. But one of the questions that came up when we very first found that West Nile virus, that we were able to detect the presence of West Nile virus in wastewater was, is it actually from humans, or are mosquitoes contributing to the wastewater? And so sometimes mosquitoes, they try to get into it's in comparison to 120 or 115 degrees in Phoenix, mosquitoes might prefer to live in a sewer shed or in stormwater sheds. And so we wanted to know, is it mosquitoes contributing to the wastewater or humans? And we can't necessarily disentangle them both from each other, but we can look to see if there's mosquito DNA present in the wastewater at the same time that West Nile virus RNA is present in the wastewater. So for that, we carry out something called an invertebrate cytochrome oxidase analysis. And that allows us to look for the presence of any invertebrate species that could be present in our sample. At the same time, we also use sort of a positive control. We use a vertebrate cytochrome oxidase analysis, knowing that human DNA should be present. So it's a bit of a positive control for us. And then we also carry out West Nile virus droplet digital PCR. RT-qPCR is usually our first pass on things. West Nile virus ddPCR tends to be a little bit more sensitive than qPCR, and my group does not use digital PCR itself. We only use droplet digital. So We looked at 6 wastewater samples that tested positive for West Nile virus when we used RT-qPCR. And we looked for both the vertebrate and invertebrate cytochrome oxidase, so invertebrate and then vertebrate. And what we found is that this one is really hard to see, but it's sort of there. We found a signal for the invertebrate cytochrome oxidase in every single one of our samples, but we have to look further to see, is that invertebrate actually our target vector species, or is it a worm or something else like that that could be circulating in wastewater? And then, of course, for all of our samples, we did detect vertebrate cytochrome oxidase, which is exactly what you would expect from wastewater being contributed by humans or any other vertebrate species. After we do that, that amplification, we are able to, so we sequence that amplified product that I just showed you on the gel. And then after we do the sequencing, we align all of the sequences to West Nile virus vector species genomes. So the Culex quinque and then the Culex tarsalis. And of all the samples we tested, of the 6 samples we tested, we only found that 1 sample had reads that mapped to the vector of interest. And in that 1 sample, 31% of the reads aligned to the Culex species mosquitoes. But in 5 of the samples, we were not able to detect Culex DNA within the wastewater sample. So given that West Nile virus was circulating, that there were West Nile virus positive human cases at the same time that these samples were collected, and that we were not able to identify any Culex DNA, it sort of gives more evidence for human contribution of West Nile virus to the wastewater. And if we look at this a little bit closer, what we've plotted here are the epidemiological weeks in 2023. And in blue, the blue bars represent the number of human cases in all of Arizona over this time frame, where 72 of the 82 cases were in Maricopa County. And again, so thinking the human cases are from all of Arizona. The wastewater positive samples are only from Tempe. And what we started to see is that as the West Nile virus positive human cases were growing, we were also getting more wastewater hits. In this particular week, we got 3 wastewater hits. And that was carried out through a lot of 2023 when we were seeing human cases. The next thing that we decided to do, and this was really City of Tempe that was very motivated to better understand this, and so you have to give them a lot of credit because this really was their idea. Because of the finding of local circulation of dengue in 2022, both in mosquito sample and then in 2 humans. City of Tempe wanted to know if we would also be able to look for dengue circulating in the wastewater. So if you look at the Arizona 2022 dengue statistics, again, what you see is that there were 2 locally acquired cases, just to show you what those data look like, versus 11 travel associated cases. And then in 2024, there were no locally acquired cases in the entire state of Arizona, but there were several travel-associated cases that were present in the state of Arizona. And then you can look here and you can see the different serogroups of dengue. Of course, there were 4 different serogroups of dengue. And then the one that we're going to be focusing on is serogroup 2. And that is because we did detect we had a single dengue detection in Tempe wastewater. That was in spring of 2024. And the subtyping results using our droplet digital PCR approach was inconclusive because we didn't get a high enough number of droplets for a conclusive, yes, we definitely found serotype 2, but we did get the highest number of droplets for serotype 2, so it was suggestive. And then we talked with Maricopa County, and they did approve of us releasing this information, that the dengue detection in the Tempe wastewater coincided with 2 travel-related Maricopa County cases, one that had serotype 2 with a Tempe home address. So that made us feel pretty good about the ability to detect dengue in wastewater. Okay, so for this particular section, I have another section coming up. Our conclusions are that West Nile virus is endemic in different locations throughout the southwestern United States, and genomics can tell us where those endemic locations are. We also can use phylogenetics. Sorry, I'm trying to move my. We can also use phylogenetics to identify different important locations for maintenance of the virus within Maricopa County and other locations that have endemic populations. We're still looking at the potential of genomics to predict human case risk and trying to complete this in a near real-time manner. And then finally, wastewater-based epi can be a useful tool for monitoring human shedding of West Nile virus and dengue, as I showed you on the last slide. And we have so many different wonderful partners that participate in this work. I mentioned City of Tempe, but we also work with several different locations in the Western United States. So we're pretty proud of that. I thought what I would do, if this is okay, since it's 1:32, is stop for a minute and see if anybody had questions specifically related to this section of the talk.
Yeah, it does look like we have two, if you don't mind. I'll go ahead
Oh
and read them.
yeah.
Yeah.
Oh, go to part two.
Oh, no, I have two questions.
Oh yeah.
All right, so the first one, are there any trends of diseases happening in Texas?
Oh, trends of diseases happening in Texas. I don't specifically know. However, we do have a collaborator up here at Northern Arizona University. His name is Spencer Fox, and he has very specifically developed models of I think influenza and potentially dengue spread in Texas. Um, but we have not integrated, that's interesting. We haven't integrated any Texas-based samples into any of our analyses at this point in time.
Okay, thank you. And then the second question, wouldn't it seem important to also consider density in a positive mosquito pool as another graphing parameter in relation to case, location, and geographic density?
Yeah, let me write that down really quick.
I can repeat it as well if you'd like.
Oh, I just want to make sure that I can specifically look into this later as well, but I do have an answer. So consider density of mosquito pool. So yes, I definitely think that it is important to consider density of mosquito pool. One of the things that can be a little bit difficult when carrying out RT-qPCR for viral detection is that you need to have a well quantified positive control because RT-qPCR just gives off fluorescence. And so you can say, well, this particular mosquito pool was, very, very positive versus this pool was less positive. So you can do sort of relative comparisons, but you can't actually do numeric comparisons without a good positive control. And so my group did recently grow up and then deactivate a virus that we provided to the county. So that in the future, if they do specifically want to do relationships between number of mosquitoes in a pool and the absolute quantification, that they can do that. But at this point in time, I think that most of the vector control agencies are not taking absolute values into account. Yeah.
So that seems like the last question, so you can go ahead and move on.
Okay, great. Thank you.
Thank you.
Okay. Oh, and this, so I'll leave this up for just a second. So the different visualizations that we put together, so those maps that move over time, if you wanted to use your cell phone and take a picture of this, that would lead you to a website that would show the phylogenetic tree that we made. And then also next to it, you'll be able to see the map. The map and how the different West Nile virus variants are present on that map. And if anybody has a question about how to use that, please email me and I'd be happy to fill you in on that. Okay. For the next part of the talk, we're going to get away from West Nile virus and talk about additional wastewater surveillance in Arizona. So a lot of this work was done at TGen North, and it focuses on EVD68. So just to give you a breakdown, these are all of the different targets that we look for in wastewater very routinely with our different collaborators. At the bottom, we have something that we call these semi-agnostic pan assays. Agnostic means, in this case, semi-agnostic means that we're not only looking just for one particular pathogen, we're looking for a whole family of pathogens. And so we use a PCR approach that is a little bit more general and can amplify all sorts of different viruses. And then we use sequencing to classify which viruses are present in a particular sample. And we have that for a variety of different viral families. And then, of course, I showed you we already do that for vertebrates like birds with the mosquito blood meals, and then for longer microbiome-based analyses as well. But this talk is going to really look at EVD68 and actually not dengue surveillance because I moved those slides up. We have several different wastewater biosurveillance partners, and the group that I have not included on here yet because it's a fairly new collaboration is we're also processing samples for the New Mexico Department of Health. Right now, specifically looking at arboviruses and really focusing on arboviruses. When we look at the city of Flagstaff and how the wastewater treatment plants are set up, we really have two wastewater treatment plants. We have the Rio de Flag where the blue X is, and then up here we have the Wildcat Hill wastewater treatment plant. And my understanding is that it takes about, if anything has to be discharged from the Rio de Flag plant, it would take 3 hours for the discharge to get up to the Wildcat Hill wastewater treatment plant, which is a larger plant that can handle more product. But really, the vast majority of our 86001 zip code and ends up in the Rio de Flag wastewater treatment plant. 86004 will go into Wildcat Hill, but sometimes we will get some of the extra that Rio de Flag doesn't process, that'll end up coming over to Wildcat Hill as well. So you can expect sort of a mixed signal in this top right area, and then a more zip code resolved signal at this blue X. And then also we were doing a lot of sampling at Northern Arizona University. And if you look at the map of Northern Arizona University, you see all these little green dots, and those little green dots represent all the different manholes that were potentially available to sample on campus. And we worked with our administration when we were processing wastewater NEU previously during the height of the pandemic to figure out what sites would be best representative of the population, of the particular population. And then, so more recent partnership, this is August of 2022 is when our collaboration with Tempe started. And so here is a map of the different Tempe wastewater sewer sheds. Really, they're called the BioIntel basins in city of Tempe, and they put a lot of work into setting up locations that they would be able to sample from and really get a good idea of population health for the city of Tempe with regards to wastewater-based surveillance. So what we're going to focus on for this part of the talk are enteroviruses. And so the most notorious enterovirus, of course, is polio. And luckily, there doesn't seem to be a whole lot of polio circulating, thankfully. But there are other outbreaks that happen every single year. So we'll see EV-A71 that causes hand, foot, and mouth disease that can really be problematic, especially in daycares, little kids touching each other, and they end up with rashes on their hands, feet, mouth, and sometimes other places as well. And then this other virus is called EVD68. So EVD68 is a non-polio enterovirus that can cause neuroinvasive issues. Usually it's just a respiratory, it mostly causes respiratory issues, sometimes some gastrointestinal issues, but very rarely it can cause acute placid myelitis. And on the right-hand side, this graph is data from the CDC, and you can see that the first time that we really recognized AFM in the United States, which can cause partial or near complete paralysis for a period of time. You can see that we saw several several cases in 2014 and then in 2016 and 2018. So just re-emerging every other year. And then, of course, we had the pandemic. And so the number of AFM cases was down because there was a lot of social distancing going on. But in 2022, you can see there was a little bit of a blip. And so the blip is what we're focusing on. My group, what we did is this is back to the semi-agnostic pan-viral analysis. And what we did is we developed an assay, or we optimized an assay that was already present, focusing on this 5' UTR region of the enterovirus genome. And it produces an approximately 500 base pair amplification product. And so when you run it on a gel, you know that a sample is positive for that amplification product because you see a band coming up at about 500 base pairs. So this is a variety of different wastewater samples. Then we sequence this amplification product, and we can compare it to a database that's full of different enteroviruses and rhinoviruses and start classifying which enteroviruses and rhinoviruses are actually present. And you can see, and I have to tell you, we were incredibly surprised, a whole lot of green. And that green in this case is EBD68. So then we wanted to know a little bit more about EBD68, how it was circulating throughout the population. So we developed something that's called a whole genome tiled amplicon primer design that would allow us to sequence the entirety of the genome. And this down here just shows how well the sequencing worked. You can see that there are some regions that drop out, but generally we're able to get good sequencing product from this analysis. And when we looked across City of Flagstaff congregate living sites, City of Flagstaff wastewater treatment plants, and City of Tempe biointel basins, you can see again, there's quite a bit of green. And but in this case, so the colors change on this slide, but so in this case, the green is Coxsackie virus A6. But the orange color that we're seeing, that's enterovirus D68. And so that was, again, the one that we were really surprised to pick up, primarily because we were using an older assay, and that assay was not working to pick up EBD68 in the wastewater. There were a lot of other enteroviruses that were present as well, but again, EBD68 is what we're focusing on. What we decided to do, because we failed to initially detect EBD68 with an RT-qPCR assay, and then we missed it until we did later sequencing, is we developed a brand new RT-qPCR assay. And what you do when you develop these assays is you start out with a positive control where you know absolutely the number of copies of virus there are in that control. And then you look to see how low you can go for detection purposes. And this is a little bit backwards, but a lower CT value means better detection and a higher CT value means lower detection. And usually we only consider a sample to be positive if it has a value of less than 40. And so then you can see that at some point in time, you do lose detection. And this 1,000 copies per milliliter is the same as one viral copy per microliter. What we also have to do when we're validating these types of assays is we have to ask the question, are we able to just pick up EBD68? Or is it also going to pick up closely related enteroviruses? So you can see that we tested out our assay versus a lot of different EBD68 variants. This one right here, it didn't work on, and that's because that variant was collected in the 1960s in California. And so generally it's quite, from an evolutionary perspective, it's quite different. And so most tests do not pick this one up. But what you can also see is that it doesn't pick up on EBD 111, EBD 94, or EBD 71. And so this particular validation makes us feel pretty good about the assay that we developed. We also compared our assay performance to the performance of a CDC assay that's older. Now they've also come out with a new assay. But the older CDC assay versus our assay, you can see pretty easily that there was less detection in the older CDC assay, but the new one is quite good. Okay, so then we wanted to know, is there any connection between, we wanted to be able to compare the assays to each other, compare Flagstaff versus Tempe, and then we also wanted to compare the wastewater results versus human case counts. And what you can see in this first panel is that we looked at just the CDC 2020, the newer 2022 assay. And you look in Flagstaff and Tempe, and you can see that EVD68 in the wastewater is pretty similar in both locations. So whether you look at up north or in central Arizona, we look to be seeing the same trends in those locations. And that was also true for the our D68 detect assay that we developed. And then if you look here, down here, and you're comparing the CDC 2022 versus the D68 detect assay just on Flagstaff wastewater, you can see that the new CDC assay and our assay very closely mirror each other. And that's also true for the Tempe wastewater. But what I think is really interesting about this particular slide is that in purple, what you're seeing is from our collaborators at Northern Arizona Healthcare, they don't specifically test for EVD68, but they have a pan assay that will detect if a sample is positive for any rhinovirus/enterovirus. And what you can see is that, okay, so they had some detections for rhinoviruses and enteroviruses here, but then all of a sudden you see really that the number of enterovirus, rhinovirus detections really starts climbing. And again, you can't attribute this to any particular virus, but what we do know is that our enterovirus D68 wastewater assay showed climbing of EVD68 at the same exact time that there was increases in the human cases. And so it really does look like EVD68 is probably what was causing the majority of human cases that we were seeing in Northern Arizona Healthcare at this particular time. And so it also you can see that the wastewater signal in the black and gray tended to come up a few days in advance of the human cases. What we're able to do then is after we can do genomic sequencing, and enteroviruses are pretty diverse, but the enteroviruses, the EVD68 is pretty diverse. But B3 tends to be the one that we see circulating the most in the United States. And in the orange, you can see all of the different genomes from our study in City of Tempe and City of Flagstaff. And if we zoom in on the City of Tempe and the City of Flagstaff samples, you can see that there is at least one other so City of Flagstaff location that doesn't cluster with the rest of the Arizona locations taken at this time. But by and large, the vast majority of the Arizona samples, EVD68 does tend to cluster together, indicating that there were a limited number of introductions into Arizona of EVD68, but that it spread pretty quickly after introductions. So summary and conclusions for this part. We were able to identify EVD68 in wastewater after developing the new assay. We found that the viral load peaked approximately August 29th of 2022. This is something that I left out, but we did carry out our congregate living site wastewater collections using ultra-sized tampons that were allowed to sit in the wastewater for a couple of days. Okay, the clustering of the phylogeny indicates that there were a limited number of introductions into Arizona, but then rapid spread after that. Wastewater wastewater-based surveillance did detect EVD68 a few days before the positive individuals were seen at the hospital. And it really does show utility for wastewater-based epidemiology when you're thinking about what could be circulating in the wastewater. And what we do is we communicate our results to our county, state, and local public health partners, and then they are able to communicate to all of their additional partners who they feel really need to have an awareness of this data. So we have a lot of different collaborators in this area as well. Just so thankful to the Maricopa County, City of Tempe and Flagstaff, Arizona Department of Health Services. We wouldn't be able to kick any of this off without the collaborations that we have in place. And with that, I can take any questions. Thank you.
Okay. Thank you so much for your presentation. It was awesome.
Oh, thank you.
So the first question, can large bird populations in a localized area contribute significantly to the spread of West Nile?
Is that a question for me, or is that you're asking to everybody else? Sorry.
Oh, sorry, that's a question for you. It popped up right after you finished part one.
Cool. I wasn't sure if it was a poll, and I was like, oh, I know that one. Okay, so large bird populations in a localized area. Yes, it can certainly can contribute. So something that I didn't bring up, but on one of the earlier slides where I was showing human cases and mosquito positive pools, you could see that that those numbers would get higher and lower every other year, and that is really because of the bird population. So what happens in a really hot year like 2021, you'll see a whole lot of mosquitoes that are positive for West Nile virus, and those mosquitoes are biting birds. And when mosquitoes bite birds, especially the resident species in Arizona that don't usually die from the infection, they become immunized. And so you get this huge immune population of birds in one year. And then the next year, West Nile goes down because you have so many immune birds. But then those immune birds have babies, and then those babies, they're not immune. So you have a new susceptible population. So if you have a large susceptible population of birds in a very localized area where there's a lot of mosquitoes, and West Nile is able to be maintained there, it can certainly contribute to significant spread.
Thank you.
Sorry, that was long.
No, not at all. It was great. I make notes with all the answers. But I mean, that looks like all of the questions. Someone commented that was impressive data. I do agree.
Thank you.
So with that said, I mean, do you have anything to add or you are all set?
As far as I go, I think the only thing that I would say is that I've given different variations of this presentation on different to different folks, but my group really does enjoy giving talks to our public health folks. But if there is anything that you think that we can improve upon, or something that you think that anybody thinks I didn't describe well enough, or different questions that you might have that can contribute more to the research, we're always happy to incorporate that in, because we're always trying to grow this project so that it's meaningful research that can assist public health purposes. So I just wanted to throw that in there.
Well, thank you so much. Really
Thank you.
appreciate your time today.
Yes, thank you. Bye.
Okay, so it looks like we ended a little early, so I'm gonna go ahead and share some of the cottage food videos. So let me just go ahead. Share this here. All right, give me a thumbs up if you can see my screen. Great.
Looks good,
Thank you,
Rebecca.
guys. Thank you. Okay, so this is our cottage food guidance page. We have our food safety video series here. It is just like a YouTube playlist, honestly. So I'm just going to go ahead and click on one that I think is interesting. We'll probably just go to the food safety handling, so that's kind of what we're working on with today. Let's see here. Cooling food safely. And this is what we're going to be sending. This is what our cottage food registrants will be looking at, or aspiring registrants. So this is, I mean, it's pretty basic, probably common knowledge for a lot of our health inspectors, but just introducing it to someone who's starting off. So I'll go ahead and share that now.
Hey cottage food entrepreneurs! Cooling food the right way is one of the most important steps in preventing foodborne illness. Here's how to do it safely in just 2 steps. First, cool your hot food down from 135 degrees Fahrenheit to 70 degrees Fahrenheit within 2 hours. This helps get food out of the temperature danger zone quickly before bacteria begins to grow. Next, cool it down from 70 degrees Fahrenheit to 41 degrees Fahrenheit within the next 4 hours. That keeps bacteria from multiplying and keeps your food safe to serve. If your food doesn't reach these temperatures within the required time frames, you must reheat the food to 165 degrees Fahrenheit to restart the process or discard the food. You should always use metal or glass containers, not plastic. Divide food into shallow containers that are 4 inches deep or less. Tightly cover food after it is fully cooled. Remember, 135 degrees Fahrenheit to 70 degrees Fahrenheit in 2 hours, and 70 degrees Fahrenheit to 41 degrees Fahrenheit in 4 hours. Follow these steps every time to keep your food out of the temperature danger zone and your customers safe.
We have time for another, so I'm playing more. I'm very proud of these, and we had so much fun putting these together. So we did how to cool. Let's go to home canning safety.
Hello, cottage food entrepreneurs. Canning is a great way to preserve your products, but only when it's done safely. If done incorrectly, it can lead to risky or even deadly home canned food products. Be sure to follow these tips to ensure your customers' safety. Follow current tested recipes, like those from the USDA Complete Guide to Home Canning. You can find a free copy of the guide online or purchase one like this on Amazon. Do not use outdated cookbooks or family recipes as they may not meet today's safety standards. Using the proper canning equipment can produce a fantastic shelf-stable food product. Using the wrong method, like boiling water canners for low-acid foods such as meats and vegetables, can be life-threatening. It is important to understand that boiling water canners do not protect against botulism poisoning. This bacteria, Clostridium botulinum, can survive in canned food and produce a toxin that can affect your nerves, cause paralysis, and even death. Ingesting even a small amount of contaminated food can be deadly. The symptoms of botulism poisoning may include double vision, blurred vision, drooping eyelids, slurred speech, difficulty swallowing, dry mouth, and muscle weakness. Seek medical attention immediately if you suspect you may have botulism poisoning. Always use pressure canners or cookers when producing cottage food. Using the right equipment for the type of food you are canning is important. For instance, you must use a pressure canner for low-acid foods like salsas with onions, garlic, and peppers. Make sure your pressure canner's gauge is accurate before each use. Processing times depend on the type of food, jar size, and whether you're hot packing or raw packing. Follow the USDA chart found in the USDA Complete Guide to Home Canning exactly. No shortcuts. Never eat canned food if the jar is leaking, bulging, cracked, spurting liquid when opened, or smells bad. Discoloration or mold is also a red flag. And remember, never taste food to test its safety. If you suspect a jar is unsafe, discard it without opening or puncturing it. Your and your customers' health are worth more than a single batch. Safe canning protects more than just your food. It safeguards your business, your reputation, and the trust of every customer you serve.
Well, thank you guys so much for checking those out. I'll go ahead and hand it over to you, Jake.
Awesome. Those came out really well. We'll go ahead and we'll take a short break, just long enough to go get some water, go to the bathroom. Come back at 2:05 and we'll start our next presentation.